So, colleagues, good evening to everybody. Today, Andrey Shevtsov will present a talk as an introduction to CAFA5 Kaggle competition. This is competition to predict the biological properties of the proteins. So it is a fifth round of this competition. It started about maybe 10 years ago, and this is important research topic in the bioinformatics. So thank you very much, Andrey, that you agreed to make this talk. And let me make a few comments before the start. So first, colleagues, you are very welcome that we see many participants on this meeting. And if some of you want to share his experience in this kind of meetings, you are very welcome. Please contact with me or other organizers, and we will schedule your talk. It can be short talk or longer talk. We are very informal. Please don't hesitate. Also, if you know some other person who can share his experience, for example, there are many research papers. If you know some of the authors and you can contact with them, so please do it. Another thing, as I said, we are quite informal. So we are welcome to ask the questions during the talk, but we have some small problem. Sometimes the people called Zoom bombers come to our meetings and they make noise and try to kind of spoil the meeting. So that's why we not allow you to unmute yourself during the talk. So please, if you have a question, don't hesitate to write it in the chat. I'll see it and I will spell it out for Andrey. And we'll have, after the talk finished, we'll have a free discussion. Okay, so Andrey, thank you very much again, and please start.
Okay, thank you, Alexander. Yeah, feel free to write your questions. Today I'm going to tell you some briefing introduction about CAFA, about biology and some basics of CAFA competition. So my speech is directed to my aim is to give you some basic biology information about the inputs and targets of the competition, and then say some words about data sets. And then also I will mention, as I think important part, is similarity analysis of proteins or other biological sequences. And then I say some words about language models and about 3D structure prediction, and a couple of words about the competition metric. So this is our plan. Okay, about proteins, a very basic information. So proteins are biological polymers, polymer molecules. They consist of monomers, which are called amino acids, and proteins play a big diversity of roles in cells. So they are an important target of many researchers, and have a great potential for many biology fields. So proteins have four stages of structure, for for stages of structure. And the primary structure, it's a sequence of amino acids, and then this sequence is folded into some patterns. These patterns, this folding structure is called secondary structure, and then these patterns are folded together, and they form a 3D spatial structure, which is the widely known structure of proteins. We usually see the proteins in this tertiary structure, and there is another structure which is called quaternary structure, and this occurs when multiple sequences, more than one sequence, folded in 3D in tertiary structure. When they unite, so we obtain a quaternary structure. And a couple more words about every structure of protein. So the primary structure, as I said, it's a sequence of monomers, which are called amino acids. These amino acids have one. They are usually divided on two parts. First part is called backbone, and second part is called side chain or radical. And the backbone consists amino group and carboxyl group, which are this is printed at the middle of the slide, and here is a side chain. So this side chain, we have about 20 of amino acids, and they are they differ by this. They have exactly the same backbone, but differ by their side chains. And these side chains, they determine the amino acid properties. So at the bottom of the slide, there is a couple of amino acids that are connected by amino and carboxyl group, and they form the backbone of the protein. And we see the backbone, and the balloons with different colors are the side chains. And as I said, they determine the side chains determine the amino acid properties, like hydrophobic or hydrophilic amino acids, and this will influence how this sequence will fold in the environment. These biological sequences, like proteins or DNA or RNA, are usually stored in files with the extension .fasta, and this is a text format. which is a pretty similar text format, which is presented in the middle of the slide. It consists of two parts. The first part starts with this symbol, and this line contains metadata of the sequence. So the first code is the UniProt ID in case of proteins, and by this UniProt ID you can search for this protein in the internet or in the databases. We can also see here the name of this protein, uracil DNA glucosilase, and the host of this protein, which is Vaccinia virus. And then second part is the sequence, which is separated to 4 lines. Maybe more lines in case of the bigger length, and you can easily parse it even with simple text loaders, text readers, and also there are plenty of instruments to parse the structures, and here I show you an example with the instrument SecIO. It's from one of the Kaggle datasets, or one of the Kaggle notebooks. Sorry. Okay, secondary structure. As I already said, it's a secondary structure usually. This sequence usually form a part of this sequence of amino acids usually form similar structures, and the two huge categories of the structures are called alpha helix and beta pleated sheet. So they are presented in the right side of the slide, and usually this information, and also it is great categories, but this also can be divided on up to 8, as I remember, categories, and they are called the Dictionary of Protein Secondary Structure. They are presented at the left part of the slide, and so usually this information is stored as another sequence of letters, and you can see here the letters G, H, and so on. And this every amino acid belongs to some not every. So there are some amino acids that belongs to some letters from secondary structure. Other letters can have no secondary structure. They can be some kind of unstructured parts, which are called loops, and they are usually between some secondary parts. Here you can see alpha helix, beta sheet, and between them there is some linker part, which is usually called loop, and she has it has more conformational freedom. So next there is a 3D structure, a tertiary structure, and this sequence with its secondary parts is folded in some kind of globular structure, and to trying to minimize its free energy, and so this 3D structure or triterary structure is the common sorry. So proteins usually exist in this 3D structure. What that I wanted to say, and one of the it is also formed by side chains, by the connections of side chains and side chains of amino acids, and usually the protein is protein, as I already also said, has hydrophobic or hydrophilic amino acids, and usually hydrophobic amino acids tend to go inside the protein for less connection to the buffer, the liquid, because proteins live in some kind of liquid. And hydrophilic amino acids tend to be outside of the protein. And here I'll also show you some bone types, but I won't. I will not tell about them, not to confuse you with a lot of information. So and also multiple sequence. So sequence is usually also called chain, and multiple chains follow multiple connected chains follow to 4D structure, for quaternary structure. Okay, so there is a widespread format of storing the 3D structure, and these files which are which store the information about 3D structure of the protein have an extension .pdb, and they looks like the multiple columns separated by some space, and here we can see the the name of amino acid. So every row at this PDB file belongs to every row with the name atom belongs to the atom in this protein. There are also another called rows. They belong to other data. Usually we don't need this. We usually need only atom rows. And here we can see that with the first here with the numbers of this and not numbers. So it's the first atom. It's the atom of amino acid asp, and it's a chain name. If it's a tertiary structure, we only have one chain. If it's not a tertiary structure, there are multiple chains. And then there are the x, y, z coordinates of this atom in the space. Okay, so what I want to also mention that in this example, all these atoms belongs to one amino acid. So it's all the atoms of this amino acid. Yeah. Okay. So next. Next, some words about protein function. We can divide the protein. So proteins, as I said, plays a great role, have a lot of functions in cells and in organisms, and these functions are categorized. So we have we can divide these categories to cellular function and chemical function. And cellular function is a function in cell. Here is examples of these functions. And the chemical function is it can be named as a subcategory of cellular function. So it's a function only for enzymes, only for proteins who which react with other proteins or which are called substrates, and for these enzymes, there are two principles of coding the functions. The first is enzyme commission numbers, and the second is brand. It's improvement of enzyme commission numbers, and this is a code which is given to exact enzyme, which gives how it reacts with other proteins. And second, and about cellular function, there is, for example, a principle which is called functional categories. All these function codes, or they all have a hierarchical structure, and but in this they have some differences in the biology, in the parts of proteins which they have. So there are also two function codes based on biological pathways. Biological pathways, it's a chain of protein-protein interactions, which leads to some result, and they are usually categorized by the result of this chain of interactions. And the last one that is used in this computation, this is it's called gene ontology, and it's we will deep dive into it at the next slide. But one note. We mention genes, and I want to give you some connection between genes and the protein. So genes are parts of DNA, and it's locations where the information is encoded, where the information is encoded about the protein or some other functional element, and the process is usually follows from DNA, where we have a location which is called gene. Then this location is written from the DNA to produce some buffer element, some temporary element RNA, which is then produced, which is then also written to produce the functional element protein. So we have a correlation between the gene and protein, and so we can talk about gene ontology, keeping in mind that we that genes don't have any function instead of keeping the information. They give information which is processed to the protein, and the protein starts to do some things in cell. So we talk about gene ontology or genes and genomes, but we talk about the functions of proteins. Okay, and gene ontology. Gene ontology, it's also a hierarchical structure. It's directed as a cyclic graph with encoded functions of proteins. It contains three subgraphs or sub-ontologies, which are called molecular function, biological process, and cellular component. So cellular component belongs to the localization of the protein, and other two belongs to the exactly the function of this protein. And these codes have the root codes, so it's root codes, and then they are spread to child to the child up to the leaves, and this annotation, this functional annotation, have two types of sources. First is experiment. It can be obtained by experiment or by some computation, some prediction. And in this competition, in CAFA, they use only experimentally obtained gene ontologies, and they took it from the database, which is called UniProt KB. They also give us the release of this the database. So here sorry, yeah, we will talk about it here. So here you can see the examples, or one example of the structure of this ontology. Here is example for transcription DNA templated. Sorry, it's a example for one of the proteins. He has this. It's a part of this graph. Here we can see that there are different kinds of connections between these between its child and parents. The most widely spread connection is called EZ. It's a collection of belonging to the parent function. Also, secondly, spread is a part of. So a part of, and then is another connection is called regulates. It's a connection which is the which gives us information not about the which gives us information about the regulation, the interaction between separated functions. So one function can lead to activation or some kind of inhibition or other regulation of other function, and this regulates connection gives us such information. And we must understand that we here if we have some child function, if a protein has some child function validated, obtained, so it also has its parent functions which are connected by is a or a part of. So here at the left part of the slide, if the protein has function 1 or function 2, it also has function 4, 3, and 5, but doesn't have function 6 because it has the connection regulate. It's called a true path rule. Also, I give the tool to search for the gene annotation, which is called quick cure. Also this we can there is a great database with a lot of information about the protein, as I think it's the biggest database with a lot of information about the protein, which is called UniProt, and also the examples, the sets of for train in this competition are taken from this database, and this database has two parts. Also, it has there are two kinds of entries in this database. So the automatically added entries, which are called UniProt, and manually entered and reviewed entries, which are called SwissProt. They have this symbol, and usually researchers took only SwissProt part because it has less noise and more. It's a more reliable part of the database, and so I also wanted to share this database. It has the interface to search, and we can take we have this FASO, for example. Here there is, as I mentioned already, a second. Here there is a UniProt ID, and we can go to the database and search for this protein, and here we can see its name, function, and even its geoontology. So here it is, and we go further. Okay, so about the data in competition, there is a file geobasic with extension .oba, which has all the terms that are in the competition. It can be opened and visualized with the OBANet tool, and there are notebooks with these manipulations, and the main file is called train terms. It's a table, and this table has three columns. Entry ID, it's a column with uniprot IDs of the proteins. Then the term, it's a column with these geoterms, and the column aspect, which belongs to the categories of the of these geoterms. And here at the right side of the slide, there is the distribution of these aspects. So we can see that the most part of the data sets are biologically processes aspects, and the least part of the data sets are I forgot the MFO, and the second part is the cellular location, cellular compartment. And another some information about the sequences in datasets in this competition. So here is the distribution of protein sequences lengths. If I correctly remember, this is for a train set, and the mean of the lengths for the sequences is about 300-400, and the maximum length is bigger or near to 3,500. But mostly 99% of the data have the length less than 300 amino acids, and we have near to 1.5 sequences in train set, and similar count of sequences in test set. And also, I want to mention that some of these sequences are duplicates, and also there is some intersection between proteins in trained test set. So about 35% of proteins are equal in train in test set. Also, there is a file which is called train taxonomy.tsv, which consists the information of the species of the hosts of these proteins, and there is a big diversity of taxonomies in these sets. So there are more than 3,000 species in train set, and about 90 species in test set, and 70 of these species are intersect with the train set. And the main class, the main species, the main class is Homo sapiens, and so the main class are most widely used in biology and most widely researched in biology. So it's not a great surprise. Okay, now I want to tell a little about sequence similarity. So we in biology we have an assumption that similar sequences has similar functions, and so we can, one of the ways to find the function of this protein is to find similar proteins and to watch their functions. And the commonly used similarity metric is the score of the alignment. So we take two sequences. In simple case, we take two sequences and place one under other, and place the or place one sequence by one, or place a not one letter by one, or place under some of the letters gaps, no letters, to obtain the best score of this obtained alignment. And this alignment score is equal to the sum of the scores for gaps, for matchings, match when we have exact same letters at this column, and mismatch score, mismatch when we have different letters. So there are gaps usually have a big score below 0, and it's usually hyperparameter. But match and mismatch scores have its empirical values, which are gotten from the frequency changes of amino acids. So scientists took a lot of sequences, and they search for the most commonly they align them, and then they search in these aligned sequences for most commonly used or least commonly used changes of the amino acids of these letters. And then by this frequency, they calculate the matrix. One of popular matrices is called Blossom, and here it is at the left bottom part of the slide, and there are scores for each change of the amino acids. So there are also two types of alignments. First type is called global alignment, when we want our sequence, when we think that our sequences are have a good alignment, when the whole sequence needs to be aligned to the whole sequence So whole sequence needs to be aligned to the whole sequence. If we have a local alignment, we can align only we can align sequence only to part of the sequence. So here at the right part is the global alignment. And if we had, for example, a part GAA only without left and right part, we will want to make local alignment, and we will try to find a best place in this sequence to locate this sequence. So this is evolutionary characteristics. So if we assume that proteins or the proteins mutate in time, have some changes in letters in time, and more letters changed, great evolutionary distance between two sequences. And we also, as we can see in this blossom matrix, we also have more critical changes, which have great minus score, or we have similar changes when the sequence, when the letters, when the amino acids has similar properties. The change will not give us some critical change in the protein, and we can freely change these amino acids. And so in proteins, after alignment, we can find some conserved parts, which here marked as gray parts, conserved parts which are equal in multiple sequences. And we also have some conservative regions which have some diversity of letters, and it can be if it's a low diversity, it's a conservative. If it's a great diversity or not diversity, or the changes in these letters have a great negative score, it's non-conservative part. And by these alignments, we can even make evolutionary trees of the of these proteins. And in this evolution, in these trees, in this tree structure, we also have similar proteins with similar functions at the same parts of this, at the same branches of this tree. Okay, so as I already mentioned, we can, it's one of the simplest way, one of the most, one of the oldest way to find out the protein function is to search for how this protein, how its sequence aligns to other sequence, which usually stored in some database like UniProt. But there is one problem that multiple sequence alignment, when we have multiple alignments, is has a high computational cost, and we can't use the simple methods for it. We can't make whole combination of pairwise alignments of this sequence, because it's a great computational cost and a lot of resources, a lot of time. So we need some heuristics for making this search. And one of the most popular heuristic is called BLAST. BLAST was called also as a Google of biological research. It's a fast and efficient heuristics of multiple sequence alignment, And here is presented the algorithm of BLAST. And in a couple of words, it consists of 3 steps. At the first step, we take our query sequence and cut it to little parts, which are called words. Not even cut, but make commas. So we make one stride and obtain the words with a stride. And then we take these words. Also, we add the very similar words. So we take the word, and then we can even take a very similar word to it by changing similar letters. As we, it's about Blossom metrics. It's about change letters metrics. We can change some letters that have not negative or little negative score for changing, and obtain even more words to the following steps. So then, and here is also another matrix which is called PAM. So PAM and Blossom, in most cases, one of these metrics is used, PAM or Blossom. So then we take these words, little parts of sequences, of query sequence, and try to exactly match these sequences in database with database sequences. When we exactly match this query sequence, we try to extend this alignment by other parts of the by other left and right parts of the query sequence, and we add the left and right letters until we get some threshold score. So we add match the word, and then this matching has some score of alignment, which is the sum of matches and mismatches. Here is the matches, so it's the sum of matches these letters, and then we extend it. And after every iteration of extension, left and right letters, we calculate again the score, and it can increase or decrease, and we have some threshold of this score. After we obtain this threshold, we stop this iteration and save the results. Then we have some statistical statistical evaluation, after which we do it for a couple of times, and we have some statistical evaluation. After that, we obtain the alignment score, which is the sum of the scores of this, as they are called, high segment pairs or maximal segment pairs. So, for example, I also present here the result of the usage of this BLAST search in one of the notebooks in Kaggle, and addition of this BLAST search to one of the algorithms, one of the deep learning algorithms, increases the score for a couple of numbers. So then, about language models. Some words about language models. Proteins can be treated as letters as natural text. They also have its alphabet, which consists of 20 letters and some additional symbols. And we can use architectures similar to architectures used in natural language processing. And in a couple of past years, the multiple protein language models were made. And one of the most popular models are the ESM model family, which was made by Meta company, and the ProTrans model. Both of these models, ESM model, all models from the ESM family based on BERT architecture. And there are a couple of models from 6 layers to 48 layers, and the max length of the max possible length of the sequence is 124. And ProTrans family, which has BERT-like models, and also it has a T5 model, and the maximal length of the input sequence for it is 512. So for all these models, the embeddings are already on Kaggle. And here at the bottom of the slide is example from one of the instruments for gene ontology prediction. It's a simple usage of these embeddings. So after obtaining the embedding, the authors created a couple of fully connected layers, and the dimension of output layer is same as the dimension of gene ontology graph structure. And also I want to mention here that all these models have a simple tokenizer, which is as simple as one hot encoder. So we used to think that for a language model, usually there are some kind of pre-trained tokenizers like BPE, but here at this there are also some models with more complex tokenizers, but occasionally the most popular one have a simple one hot encoder. So it's a screen from the discussion of this competition with the discussion about the highest possible score obtained only by a language model sequence encoding. And here the authors gives us some insights about the scores, and we can see that it's a pretty big score for these competitions, 0.5 53 54. So in my opinion, the word embeddings, the sequence embeddings will play one of the important roles in obtaining the best solution in this competition. So, and some additional information. 3D structure, we had some talk about this about 3D structure earlier, and I wanted to say about it because I want to tell about possible ways of obtaining about the possibility of obtaining not only sequence but also a 3D information in the possible solutions. So usually there are two. Again, there is two categories of obtaining information: experimental and computational. And also, experimental information is the most valuable, the most reliable information. Usually 3D structures of proteins obtained by X-ray or by cryo-electron microscopy. And there are databases with the like PDB database with the structures obtained, and you can get the experimental structures from these databases, but sometimes you don't have the information of the protein. In the case of our competition, you can not have the information, the name of this protein, or even there is no experimental 3D structure of this protein. And here we can go to some approximation to predicted 3D structure. And as I think all of you heard about AlphaFold, and second AlphaFold, and here is at the bottom of the slide is presented the structure, the architecture of these models, and it gives us an opportunity to obtain a 3D structure from the init sequence, and at the inside of this architecture, AlphaFold uses multiple sequence alignment. So they create a multiple sequence alignment of the input structure, of input sequence, and they also by this. So they inside they find similar sequences. And they also find by similar sequences, similar structures, similar 3D structures. And there are two main roads of this model. One is MSA representation, and second is pair representation. And this pair representation contains a structural representation. So it's a structural correlation between the pairs of this protein. And one of possible ways to add the structural information to the as features to these competitions, or solving some tasks with proteins if you have only input sequence, is the obtaining the pair representations or some other in some other hidden layers from this model. And also I want to mention that there is a analog of the AlphaFold, which is called OpenFold. It's open code library, open code model, which was trained with the same parameters as AlphaFold. So they copied the AlphaFold, but make it free to use. And it can even be run by Collab. So it's OpenFold. Here is the link to it. And also ESM family has its own model to predict 3D structure by sequence, which is called ESM fold. And it also has some hidden representations which have, but this model differs from AlphaFold models by the fact that it doesn't get similar structures, similar sequences. So it is more de novo algorithm than AlphaFold, but it has less accuracy than AlphaFold. But it's more de novo. And also there are for these tools, for these methods, there are already created data sets with 3D structures of proteins, with predicted 3D structures of the proteins. So, and the last part of our discussion refers to the evaluation metric. And I can't say a lot about this. I share with you the screen with the states of obtaining the evaluation metric of this competition, and take you along with these steps. So that's all I wanted to say. If you have some questions, maybe we can go to some discussion.
Andrey, thank you very much. So, colleagues, now don't hesitate to ask questions, comments, or suggestions. So we have already a question in the chat. So let me read it. So Daria asks, it's about BLAST, the slide with the BLAST. Andrey, can you please go?
I can't find the question about the BLAST. Can you please read it?
I don't. Please go to the slide in your presentation where you discuss the BLAST.
Okay.
And the question is, I don't really understand how the surrounding is checked at the stage 3. So this is about this stage 3, and the surroundings of AA is checked. So Daria, if you can.
So we have okay. I will try to answer what I understand. So we have a part, this word, which is a part of this query sequence, and when we find the matching place at the target sequences of the database, we can take from our initial sequence right and left additional letters. And we spread by this additional sequence, by these additional letters or amino acids from the query sequence. So if I don't understand your question, please reformulate it.
Okay, colleagues, now you can unmute yourself by yourself. So if you have a question, you are welcome to ask it by voice, or you can write it in the chat, up to you, or any comments, suggestions, whatever.
Hi, this is Bruno. Thank you, Andrey, for the presentation. Really clear and very interesting. I have a question. If you can go to the next slide. When you talk about, yeah, exactly, this using proper embeddings and then using basically a classifier there where the output or the number of classes is the, I guess, number of terms in the gene ontology, which I know are in the order of thousands, right?
Yes.
I'm
yes.
surprised. I'm surprised that, I mean, I haven't looked in detail into this model, or I'm just basing my question on what I see in the screen. So does this classifier work well, even though the number of output classes is so large? Because usually, you know, I would say that, you know, the number of samples that you have to train, you know, like proteins is not that they are, like these terms is not that they are highly tagged, right? So I was wondering, yeah, how that works.
Okay, yeah, thank you. So I basically don't also know the results, the metrics or benchmarks of this exactly model. And but if they share it, it should have some kind of computable metrics, and yeah. Also, I agree with you about the problem with the number of labels or classes here in the task. So and also there are a number of methods which are somehow decrease the dimension of classes, like with the convolution, like with the graph convolution to these labels, and then the convolution after the prediction. But here they use the simple approach. But as they show it, they also get some results.
May I comment on this? So, as far as I understand, these protein language models, they were not trained for this gene ontology task. So this is a kind of unsupervised or semi-supervised, like BERT, when you just take the sequences, mask a part of the sequence, and try to predict it. But it produces this, what is called embeddings. And embeddings can be used as to predict, for example, gene ontology or any other task. So this embedding is universal. It was trained without knowledge of gene ontology, without knowledge of, I don't know, enzyme classes or any other kind of task, only looking on the sequences. But they appears, it's a kind of miracle that they appears to be kind of quite good for prediction of the many different tasks. One of them is gene ontology. But going on to your question, Bruno, so what I see in the public notebooks of the current Kaggle competition, most of the people, they choose only about 1,000 to 2,000, something like most frequent terms of the gene ontology. So now in total, they have 40,000 gene ontology terms. But no one tried to predict them. Everybody takes something like 1,000, 2,000 and try to predict only those most frequent. Maybe if someone will be able to predict all the 40,000, he will kind of get a great result. But for the moment, we don't see, at least at the public notebooks, this approach.
So I duplicate my part of speech that there are some methods to reduce the dimensionality of the labels, which are also already used for this task, but not in this competition, maybe in the public part of this competition.
I guess also one thing would be that I guess the embeddings are published, but maybe you can get better so the embeddings are published, and then you are training this classifier, but maybe results could be improved if you actually do the trunk, like you actually grab protbird and allow the layers to train layers of protbird, right? To like, of course, it's computationally more expensive, but probably you will get more better results, right?
Yeah, and also there is one note that we don't even know what layer of this encoder should we use. So there is a couple of states like Bertology means biology, which gives us some information which compare the capability of different layers in the encoder to make different tasks. And there is some assumption that some layers have some exact information, like some layers have information more structural than maybe the later layers have some functional information, but it's also a field open.
Interesting. Is this what you are talking right now? Is this in a paper, or this is something you read?
One of the popular paper called Bertology means biology.
Bertology means biology.
Yes, it was written a couple of years, 2 years ago, and here the encoders are they had the maximum depth of encoder about 10, not 10, maybe 14 layers. So and also there is some information about some of the research in this field also is in one of the papers from ESM, but I don't exactly remember the name of the paper. So you can also see the papers of ESM.
And last question. Those numbers that you published, 0.5 performance, is what this notebook is reporting for this model. So there was a 0.55 of that.
Here.
What's that one? Yeah. Oh, okay. So this 0.5 is when you use MSA or BLAST to.
This 0.3 is when you add to it's a notebook where a man took spore GO, one of the public notebooks latest with spore GO. I can't exactly remember the name, and he added BLAST. So yes, he added a simple multiple segments alignment, heuristics of it, which is called BLAST, and he get some improvement of the score. Not the biggest improvement, but improvement.
Thanks.
Yes, so for instance, this 0.501, it was application of some quite recent published method. That's this Michael Tinty, who was at the beginning of the webinar. He was here. So he published the first notebook, and then quite quickly another participant combined it with his own BLAST-based method, but his own, I think, as far as I understand, and get this improvement.
As I understand, it's his library.
Yeah.
Not exactly his method, but his library. Here it is.
Yeah, okay. So this SPR of this was this 0.5. This is some new recent method published by some team. I think there are references in the notebook, and there is this small addition. Yeah. So this MT, this is Michael Tinty, who was here.
Yeah, here he is. Yeah. Prof.
Yes, so this is a GitHub page. Maybe you can open it, please.
I also I forgot that I had the additional 2 slides, and it was also on my slide. So they had a language model, and also they had some multiple alignment search. And Alexandra had already compared some tools for multiple alignment. And as I remember, Diamond was in this pack of tools.
Yeah, so Diamond is a kind of BLAST, but somehow faster. I don't know the details, but it's claimed to be 10, I don't know, sometimes 10,000 times faster than BLAST.
Mm-hmm.
I don't know how do they achieve it, but the claim is, and it's really fast. There are notebooks on the Kaggle with the application of the Diamond. So the first one was published by Елизавета Geraseva, who is not here, but if someone has a question, I think we can ask. Yeah.
So I also add that pretty simple architecture. Some complications, but generally pretty simple architecture, and it's one of the latest model in this task.
Yeah, it's one of the latest, but unfortunately, it doesn't give a very top result. So now on the leaderboard, many participants got better results. But who knows?
Yes.
It's a preliminary leaderboard. Maybe their method is better and everybody should come. But for the moment, it's not the best score.
Yeah, but we also kind of keep in mind some blending or assembly strategies which are useful for Kaggle, but not very useful in real life or even in the research field.
I think in this particular task, many researchers use also this ensembling or blend. So they take several models and average the predictions, maybe clever average the predictions. So in this particular field, this is used.
Yeah, and it's as I think it occurs with the existence of a lot of non-ML biological tools or statistical tools. So there are a lot of tools which you can't even can't even add in another way.
Yeah. So in other questions, suggestions, comments, Song, maybe you have some comments or suggestions, or especially those who are now on the competition, maybe can make a comment or something.
So one of Zong wrote that he suggests the score can be higher than 5055. Yeah, maybe.
I think, yeah, just purely on this language model, you can go higher when you combine the predictions. That's just my observation.
You mean the combine the like assembling or blending? Yeah.
Yeah.
Okay. Yeah, maybe. I didn't see the assembling of different embeddings, different language model embeddings. It's simple, but maybe I've missed these notebooks. But maybe it also gives some results.
Yeah, I think it's possible. I don't know. I am trying to speculate how the new high score has been obtained. 0.6.
Actually, I'm pretty sure it's possible to achieve about like 0.55. So I'm able to achieve like that score purely on the protein language model.
I agree. 0.55.
Mhm.
Yeah, so like my highest score purely on the protein language model is about like 0.549. So it's pretty close to 0.55.
Interesting.
For your 5.549, is this on one model or multiple protein language
Just
model?
one model, just
Just
no
one
no
model.
like one model for each aspect. So for each namespace.
Mm-hmm. So like for just maybe Pro T5, you get 0.55. Okay, that's interesting.
So I guess just a sec. So one question is that these numbers that we see on the leaderboard, I think they are basically like 50 or 100 proteins. It's very easy, I think, to overfit to a small number of proteins.
Completely green.
So
For me, it's a very painful question. I don't know how for the others.
I mean, I think it's fair to say that it's probably between 50 and 100 proteins that we are
I
testing.
think it's more than 50, but I think it's not less than 200. I made some estimations, but you can just submit some, yeah. And I made some simple experiments that I took some public leader notebook, and we see the score for this. I just copy and edit it and run and submit and several times, and I always get the different results. And for me, for some of my models, which are neural network, it's the same. So I take the neural network, I rerun it, just kind of, neural network is a little bit random. Local score doesn't change much, but on the leaderboard, you see a big change. So I think, yes, it's a big problem with
I'm willing to bet it's 100 or less.
yeah.
I mean, you can. There was some discussion of somebody creating a data set that improves the score somewhat. It just improves it, I think, way too much.
Yeah.
But whatever. So at any rate, it's a small number, much smaller than I guess it should be, or it's somehow not enough information. My feeling is that overfitting on the leaderboard is sort of nice, but it is misleads somewhat, I think.
What should we do? Does anybody have some suggestions, Song or Quinchen Lee? I know.
So for the worry about overfitting on the leaderboard, you can actually provide like your own prepare your own test validation data sets by using the new GOA file. Like just using the way they're trying to evaluating trying to select those test protein. So for example, the correct training term come from the GOA file come from late of the last year, and currently there's like several new version, so you can like choose one version and select limit knowledge protein and non-knowledge protein as your validation set, and choose a newer version GOA file and select them as a test set and try to monitor your model's performance on those data sets. At least my observation, there's like high correlation between the performance of my test set on the leaderboard.
For my models, I do not see good correlation between so maybe you make some good.
So I'm just curious, how do you prepare your like validation set and testing set?
So initially I just took a random. Now I created a notebook with a more kind of sophisticated scheme, but initial test I made a random, and on the random I see not a good fit with the leaderboard. So now I want to use this more sophisticated notebook, which is public. So I just look on the sequences and put into the same folds which are similar sequences. No standard group-wise validation.
And you obtain the similarity by the multiple alignments?
Yeah, precisely by Diamond, this package. So Diamond is so fast that it can align all these trained proteins against all the others. So probably it's not so good alignment as BLAST, or I don't know, but it's very fast. It gives you just a kind of pairwise for each protein, you know how it's aligned with any other. So you get all the pairs if they are on zero. So he doesn't give you not, if the proteins are very far from each other, it just doesn't return you them, but it returns you only those which are not better aligned. Of course, Diamond and BLAST, they are kind of approximate algorithm. This is not a perfect alignment. So
yeah, maybe another variant is to calculate the distance in embedding space.
Yeah. That's an interesting question, how the distance in the embedding space corresponds to the distance in the alignment.
Yes.
I made but I didn't finish these investigations. So I see it's more or less matching each other in some simple cases, but I did not make a deep investigation.
I wanted to ask, is anyone here currently fine-tuning? I mean, maybe a smaller model, because yesterday I implemented something which I can share also next days, where I fine-tune the smallest ESM model and runs also on Kaggle, but I'm not sure if I want to go down this path because I have limited resource and it takes quite a lot of time. I'm not sure how long takes one fold, but yeah, so if anyone did this.
So you've fine-tuned the whole encoder, yeah?
Not the whole. I froze most of the layers. I took a notebook, another notebook from another competition, and there the approach was to freeze everything but the last 3 layers, and this works. I mean, let me take a quick look, then I can tell you also how long this takes. But this worked in a reasonable time, but I'm not sure if the small model is enough. When I took the second smallest model, I got, after 1 epoch of fine-tuning, I got 0.37, which is a little bit low, but let me take it.
It's to the question of taking the layers of the encoder, what layer of the encoder we should take, and I think that I maybe can one of the ways to make it is to embed is to get not I've created a dataset with only the last layer, but there is an opportunity also to get all the layers from the encoder, and then take embedding from the exact layer, and then add layers what you want, even with the ways of the ESM model.
Yeah, I mean, what you mean? You mean to get the output, for example, of the second last layer and combine with the last layer, or?
No,
or do you really mean fine tuning and just unfreeze the?
I think it's equal cases.
Yeah.
For me, it's equal cases. So it can be. So you don't, as I think, you don't need to run your model with ESM or ProTrans while training. You can just take some layer. You've just taken the third last layer. So maybe it will increase the opportunity of the resources.
Yeah, I mean, that's what I actually I was a little bit surprised that even I could fit it in a reasonable amount of time. So for the very small model, I could fit, and unfreezing the last 3 layers, I could fine tune one fold in like 40 minutes. So that's
it's not wrong.
I'm thinking maybe it's good to take this for further ensembling, to take one of the smaller models, but I'm not really sure if this will work. But I mean, I guess it's worth trying out, right? Because I'm feeling with the embeddings, I mean, you get to a point where it's very difficult to improve further. I mean, that's what I
maybe.
But I don't know.
So, and you have a public notebook, yeah? Or
I can make this notebook now. It's a little bit messy, but I can share it.
I think it will be good if you.
I mean, it's running already and it's working, but it's a little bit messy and not very clean code. So I think I clean it up a little bit, and then in next 2-3 days.
Okay, thank you.
There is one notebook where, but I'm not sure, do we fine tune or not? There is some. Andrey, if you can open the code section of the.
Okay.
There was, and the also the notebook something like 3 layers. Maybe if you order them by recently created, not by hotness. And then there is something, someone compares the 3.
I believe you might means like 3 different polling methods.
Yeah. Yeah.
Yeah, but I think what he did there was
I believe that person do the like mean polling and the max polling and CLS polling. So like basically that still is the last layer, just like different representation, because like normally the last layer would be L by D, where D is the embedding dimension and L is the sequence length. So to reduce the memory usage, people usually just do a mean polling through all the residue.
So you mean he doesn't fine tune? Is my understanding correct?
Yeah, he doesn't fine tune the stuff. He just like take the like last layer representation just using different like polling method.
Okay, thank you for the comment. Yeah.
Here I just finished one notebook which implements mean embedding, so if you're interested, you can take a look. This is, I think, what he's done also, but with bigger model, this smaller model.
Okay, yeah, thank you very much.
And you can also do different stuff. What I know from past competition, that you take, for example, the last 4 layers and somehow combine them and stuff like this. So maybe this also a way to go.
Yeah, I believe this way would improve the result, like by taking like more more layer as a representation, we will like conserve more, maybe more evolutionary information.
And what do you think if, for example, take different models, so this T5 and then ESM, and what's the best way to combine them? I don't know, maybe just blend the models based on them or concatenate the embeddings. I tried both, but not really kind of got some insights. And it was very preliminary.
I think, like always, it's something that you should experiment with, right? So I mean, it's it's a good idea, and I think if you the question is of how to use these embeddings, because one issue could be that that we don't have that many proteins to test on. So if you give too much information, then maybe your model will just try to remember all the proteins. So, but I think these are all really good ideas of combining different embeddings. I think one has to try all of these approaches with different models and just see what happens. At least this is what I am trying to do.
Okay.
Yeah, I think, for example, something like this here could also work. This is also from a past competition. And I was thinking now to implement something like this, which basically takes many embeddings of different models and concatenates them, and then uses this as features to the model. So maybe this can also be a way.
Yeah, good idea. Yeah, thank you.
Semen, and did you deeply dive into results of past competitions?
What do you mean by? I mean, this what I posted now, this approach, I think it worked very good. So that's why I published also this notebook. What I just published 5 minutes ago. This is a preparation step. And then here I was thinking maybe what one could do, that you take many embeddings and feed them in one of these models like SVC, for example. And maybe similar approach can work here. So I wanted to try out now in the next days. But I think this worked good in this competition. But maybe some.
Okay, I wanted also to ask about the past competitions. You've told about past competitions results, about past CAFA, as I understand, or not?
No. This is not from CAFA. This is from Kaggle competition.
From Kaggle about the GO, or from...
No, but competition which also use natural language models.
It's about, you are talking about thermostability competition or not?
No. This is something else, also a natural language, but it was multi-regression problem in natural language, and but I mean, I'm not sure if this works in biology because I don't know anything about biology. But I was thinking maybe from how it looks like, maybe it could work, but I don't know.
Yeah, it can be, I think. Okay. I was interested about I didn't search for past results of CAFA5 competitions. I was also interested in what the models won at past competitions, but I don't know.
As far as the main point is, it was before the protein language models, the CAFA4. And so now the hope is that the protein language models can kind of help
improve the okay.
As far as I understand, maybe someone can correct me. I'm not sure. Kuichang, if I pronounce correctly, you have some comment, or sorry? Okay.
Okay, sorry, my apologies, but I need to go. So
okay, thank you very much, Andrey. It was a great presentation. Thank you very much for your talk, and everybody, thank you very much for the participation, sharing your insights.
Yes,
and
thank you for hearing, for participation. Thank you for organizing, Alexander.
Okay, you're welcome.
Great activities.
Thank you.
Thank you.
Yeah, thanks.
Maybe you should stop the recording.
Yeah, okay. Just let me stop it. Where is the stopping?